Hello to you from Chem Help ASAP. This video describes the idea of validating a hit's activity by confirming its target engagement. By the end of this video, students will be able to describe qualities of actives identified through a screening campaign, list steps for confirming actives, and list methods for establishing target engagement. Let's review actives. Actives normally come from compound libraries. Actives are selected from the library based on their promising activity in the primary assay. Keep in mind that because the library may be huge, millions of compounds, each library compound is assayed at a single concentration. Based on this information, how are actives advanced as bona fide hits in a drug discovery program? The first step is often to re-synthesize the active compound. This step both confirms the identity of the compound and may allow testing of a more pure sample of the material. Note that samples from a compound library are normally stored for long periods of time, and degradation of the compound is possible. The next step is to test the compound over a range of concentrations to generate a proper concentration response curve. This step allows proper calculation of an IC50 or EC50 value. The third step is to establish that the compound is indeed binding to the intended drug target. Keep in mind that the goal is to design molecules that modulate the action of a target, affect a biological pathway, and therefore provide a benefit to the patient. Confirming target binding or target engagement is critical. There are many methods, biophysical methods, for confirming target engagement, and each method has different advantages. Isothermal titration calorimetry is a method that determines the rate and energy of binding of the molecule to the target. The equilibrium binding constant is one property determined by ITC. Nuclear magnetic resonance does not measure KD, but it can give information about the location of a compound's binding to the target, which can be helpful. Surface plasmon resonance determines a compound's KD value. X-ray crystallography of a complex of a target protein and a molecule, called a co-crystal, allows visualization of all interactions between a compound and target. Of all these methods, SPR is likely the most commonly used technique because it is fast. We might have thousands of actives, and our assay for target engagement needs to be efficient. ITC is also common and relatively high throughput. X-ray gives a wealth of information, but not all target proteins can be crystallized. X-ray crystallography is also not high throughput. Neither is NMR. There are other biophysical methods beyond this short list. At this point, our active can be advanced as a hit. In some companies, the actives may be called hits, and the confirmed hit is a validated hit. Regardless, the compound will have a confirmed structural identity, a more precisely quantified potency, and evidence of target engagement. The hit is now often called a chemical starting point to support a drug discovery program. We have described the property of actives. We have described the steps required to advance an active to a hit. Finally, we have listed some biophysical methods for confirming target engagement by a hit. Thank you so much for watching. Please consider watching other videos in the playlist to learn more about the topic of lead optimization. Take care.